In the second part of the lecture about general principles of the gastrointestinal function, we are going to learn about hormonal control of gastrointestinal motility, functional types of movements in the gastrointestinal tract, and gastrointestinal blood flow. Hormonal control of gastrointestinal motility. Gastrointestinal hormones are released into the portal circulation and exert physiological actions on target cells with specific receptors for the hormone. The effects of the hormones persist even after all nervous connections between the site of release and the site of action have been severed. Table 63-1 outlines the actions of each gastrointestinal hormone as well as the stimuli for secretion and sites at which secretion takes place. Gastrin. Gastrin is released from G cells of the antrum, duodenum, and ileum. What stimulates the gastrin secretion? Uh, the presence of protein, the distension of the stomach, and nervous stimulation. What does gastrin do? Gastrin stimulates gastric acid secretion and mucosal growth. Cholecystokinin. Cholecystokinin is released mainly from the eye cells of the duodenum, mainly in the duodenum, in the presence of protein, fat, and acid in the duodenum. Cholecystokinin stimulates pancreatic enzyme secretion, pancreatic bicarbonate secretion, gallbladder contraction, and growth of exocrine pancreas. On the other hand, it inhibits gastric emptying. Secretin. Secretin is released from the S cells present in the duodenum, ileum, and ileum. It is secreted in the presence of acid and fat in the duodenum. It stimulates pepsin secretion, pancreatic bicarbonate secretion, biliary bicarbonate secretion, growth of exocrine pancreas. On the other hand, secretin inhibits gastric acid secretion. Glucose-dependent insulinotropic peptide, also called gastric inhibitory peptide. It is released from K cells of the duodenum and ileum. It is secreted by protein, fat, and carbohydrate presence in the duodenum. It stimulates insulin release and it inhibits gastric acid secretion. Motilin is secreted from the M cells of the duodenum and ileum. Its secretion is stimulated by fat, acid, and nervous stimulation. It stimulates gastric motility and intestinal motility. Although the motility effects are usually less important than the secretory effects of the hormones, some of the more important ones are gastrin. Gastrin is secreted by the G cells of the antrum of the stomach in response to stimuli associated with ingestion of a meal, such as distension of the stomach, the products of proteins, and gastrin-releasing peptide, which is released by the nerves of the gastric mucosa during vagal stimulation. The primary actions of gastrin are stimulation of gastric acid secretion and stimulation of growth of gastric mucosa. Cholecystokinin is secreted by eye cells in the mucosa of the duodenum and ileum, mainly in response to digestive products of fat, fatty acids, and monoglycerides in the intestinal contents. This hormone strongly contracts the gallbladder, expelling bile into the small intestine, where the bile in turn plays important roles in emulsifying fatty substances and allowing them to be digested and absorbed. Cholecystokinin also inhibits stomach contraction moderately. Therefore, at the same time that this hormone causes emptying of the gallbladder, it also slows the emptying of food from the stomach to give adequate time for digestion of the fats in the upper intestinal tract. Cholecystokinin also inhibits appetite to prevent overeating during meals by stimulating sensory afferent nerve fibers in the duodenum. These fibers, in turn, send signals by way of the vagus nerve to inhibit feeding centers in the brain. Secretin was the first gastrointestinal hormone discovered and is secreted by the S cells in the mucosa of the duodenum in response to acidic gastric juice emptying into the duodenum from the pylorus of the stomach. Secretin has a mild effect on motility of the gastrointestinal tract and acts to promote pancreatic secretion of bicarbonate, which in turn helps to neutralize the acid in the small intestine. Glucose-dependent insulinotropic peptide, also called gastric inhibitory peptide, is secreted by the mucosa of the upper small intestine, mainly in response to fatty acids and amino acids, but to a lesser extent in response to carbohydrates. It has a mild effect in decreasing motor activity of the stomach and therefore slows emptying of gastric contents into the duodenum when the upper small intestine is already overloaded with food products. Glucose-dependent insulinotropic peptides at blood levels even lower than those needed to inhibit gastric motility also stimulates insulin secretion. Motility Motilin is secreted by the stomach and upper duodenum during fasting, and the only known function of this hormone is to increase gastrointestinal motility. Motilin is released cyclically and stimulates waves of gastrointestinal motility called interdigestive myoelectric complexes that move through the stomach and small intestine every 90 minutes in a fasted person. Motilin secretion is inhibited after ingestion of food by mechanisms that are not fully understood. Functional types of movements in the gastrointestinal tract. Two types of movements occur in the gastrointestinal tract. First, propulsive movements, which cause food to move forward along the tract at an appropriate rate to accommodate digestion and absorption. And second, mixing movements, which keep the intestinal contents truly mixed at all times. Propulsive movements, peristaltis. The basic propulsive movement of the gastrointestinal tract is peristaltis, which is illustrated in figure 63 to 5a. A contractile ring appears around the gut and then moves forward. This is analog to putting one's fingers around a thin distended tube, then constricting the fingers and sliding them forward along the tube. Any material in front of the contractile ring is moved forward. Peristaltis is an inherent property of many cyclical, smooth muscle tubes. Stimulation at any point in the gut can cause a contractile ring to appear in the circular muscle, and this ring then spreads along the gut tube. Peristaltis also occurs in the bile ducts, glandular ducts, ureters, and many other smooth muscle tubes of the body. Peristaltis contractions in esophagus, stomach, and small intestine. As you see here, peristaltic contractions moves the content from oral to the anal site direction. Okay, contraction, relaxation. So in this way, peristaltis involves contraction and downstream relaxation in the esophagus, stomach, and small intestine propel the contents toward the anus. Now segmentation contractions. They occur in small and large intestines. Intermittent segmentation contractions in separate sections of the small and large intestines mix their contents with little forward move. Generally mixes the contents. Okay. Usual stimulus for intestinal peristaltis is distension of the gut. That is, if a large amount of food collects at any point in the gut, the stretching of the gut wall stimulates the enteric nervous system to contract the gut wall 2 to 3 centimeters behind this point, and a contractile ring appears that initiates a peristaltic movement. Other stimuli that can initiate peristaltis include chemical or physical irritation of the epithelial lining in the gut. Also, strong parasympathetic nervous signals to the gut will elicit strong peristaltis. Function of the mesenteric plexus in peristaltis. Peristaltis occurs only weakly or not at all in any portion of the gastrointestinal tract that has congenital absence of the mesenteric plexus. Also, it is greatly depressed or completely blocked in the entire gut when a person is treated with atropine to paralyze the cholinergic nerve endings of the mesenteric plexus. Therefore, effectual peristaltis requires an active mesenteric plexus. Peristaltic waves move toward the anus with downstream receptive relaxation, flow of the gut. Peristaltis theoretically can occur in either direction from a stimulated point, but it normally dies out rapidly in the oral toward the mouth direction while continuing for a considerable distance toward the anus. The exact cause of this directional transmission of peristaltis has never been ascertained, although it probably results mainly from the fact that the mesenteric plexus itself is polarized in the anal direction, which can be explained as follows. When a segment of the intestinal tract is excited by distension and thereby initiates peristaltis, the contractile ring causing the peristaltis normally begins on the forehead side of the distended segment and moves toward the distended segment, pushing the intestinal contents in the anal direction for 5 to 10 centimeters before dying out. At the same time, the gut sometimes relaxes several centimeters downstream toward the anus, which is called receptive relaxation, thus allowing the food to be propelled more easily toward the anus than toward the mouth. This complex pattern does not occur in the absence of the mesenteric plexus. Therefore, the complex is called the mesenteric reflex or the peristaltic reflex. Peristaltic reflex plus the anal direction of movement of peristaltis is called the law of the gut. Segmentation contractions, mixing movements. Mixing movements differ in different parts of the alimentary tract. In some areas, the peristaltic contraction causes most of the mixing. This is especially true when forward progression of the intestinal contents is blocked by a sphincter, so that a peristaltic wave can then only churn the intestinal contents rather than propelling them forward. At other times, local intermittent constrictive contractions occur every few centimeters in the gut wall. These contractions usually last only 5 to 30 seconds. Then new constrictions occur at other points in the gut, thus chopping and shearing the contents first here and there. Segmentation contractions you can follow in this figure again. Gastrointestinal blood flow, splanchnic circulation. The blood vessels of the gastrointestinal system are part of a more extensive system called splanchnic circulation, shown in figure 63 to 6. The design of this system is such that all the blood that courses through the gut, spleen, and pancreas then flows immediately into the liver by way of the portal vein. In the liver, The blood passes through millions of minute liver sinusoids and finally leaves the liver by way of hepatic veins that empty into the vena cava of the general circulation. This flow of blood to the liver before it empties into the vena cava allows the reticular endothelial cells that line the liver sinusoids to remove bacteria and other particulate matter that might enter the blood from the gastrointestinal tract, thus preventing direct transport of potentially harmful agents into the remainder of the body. So you see here the splanchnic circulation. So the food is absorbed except fat. Uh, Fat is absorbed via lymphatic uh, channels. Others, vitamins, carbohydrates, protein, digestion products, they are absorbed by these intestinal capillaries. They arrive in uh, liver. They pass through the sinusoids. There are Kupffer cells checking the bacterial contamination, and there are other enzymes detoxifying toxic chemicals that can enter the body via intestinal system. So, as you see, liver receives blood by portal vein and also by arteria hepatica. And then this venous blood coming from the liver leaves the liver via hepatic vein to vena cava. Non-fat, water-soluble nutrients absorbed from the gut, for example, carbohydrates and proteins, are transported in the portal venous blood to the same liver sinusoids. Here, both the reticular endothelial cells and the principal parenchymal cells of the liver, the hepatic cells, absorb and store temporarily from 50 to 75% of the nutrients. Also, much chemical intermediary processing of these nutrients occurs in the liver cells. Almost all of the fats absorbed from the intestinal tract are not carried in the portal blood, but instead are absorbed into the intestinal lymphatics and then conducted to the systemic circulating blood by way of the thoracic duct, bypassing the liver. Anatomy of the gastrointestinal blood supply. Figure 63-7 shows the general plan of the arterial blood supply of the gut, including the superior mesenteric and inferior mesenteric arteries supplying the walls of the small and large intestines by way of an arcing arterial system. Not shown in the figure is the celiac artery, which provides a similar blood supply to the stomach. Here you see the uh, superior mesenteric artery providing blood to the intestine. Celiac artery is not shown here, providing blood to the stomach. On entering the wall of the gut, the arteries branch and send smaller arteries circling in both directions around the gut, with the tips of these arteries meeting on the side of the gut wall opposite to the mesenteric attachment. From the circling arteries, still much smaller arteries penetrate into the intestinal wall and spread along the muscle bundles into the intestinal villi and into the submucosal vessels beneath the epithelium to serve the secretory and absorptive functions of the gut. Figure 63-8 shows the special organization of the blood flow through an intestinal venous, including a small arteriole and venae that interconnect with a system of multiple looping capillaries. The walls of the arterioles are highly muscular and are highly active in controlling venous blood flow. Here you see the artery, capillaries, and vein in a venous. This is central lacteal lymphatic channel. So there is a countercurrent arrangement of blood flow here. Effect of gut activity and metabolic factors on gastrointestinal blood flow. Under normal conditions, blood flow in each area of the gastrointestinal tract, as well as in each layer of the gut wall, is directly related to the level of local activity. For example, during active absorption of nutrients, blood flow in the villi and adjacent regions of the submucosa increases as much as eightfold. Likewise, blood flow in the muscle layers of the intestinal wall increases with increased motor activity in the gut. After a meal, the motor activity, secretory activity, and absorptive activity all increase. Likewise, the blood flow increases greatly, but then decreases back to the resting level over another 2 to 4 hours. Mechanisms of increased blood flow during gastrointestinal activity. Although the precise causes of the increased blood flow during increased gastrointestinal activity are still unclear, some facts are known. First, several vasodilator substances are released from the mucosa of the intestinal tract during the digestive process. Most of these are peptide hormones, including cholecystokinin, vasoactive intestinal peptide, gastrin, and secretin. These same hormones control specific motor and secretory activities of the gut. Second, some of the gastrointestinal glands also release into the gut wall two kinins, kallidin and bradykinin, at the same time that they secrete other substances into the lumen. These kinins are powerful vasodilators that are believed to cause much of the increased mucosal vasodilation that occurs along with secretion. Third, decreased oxygen concentration in the gut wall can increase intestinal blood flow at least 50 to 100%. Therefore, the increased mucosal and gut wall metabolic rate during gut activity probably lowers the oxygen concentration enough to cause much of the vasodilation. The decrease in oxygen can also lead to as much as a fourfold increase of adenosine, a well-known vasodilator that could be responsible for much of the increased flow. Thus, the increased blood flow during increased gastrointestinal activity is probably a combination of many of the aforementioned factors plus still others yet undiscovered. Countercurrent blood flow in the villi. Note in figure 63 to 8 that the arterial flow into the villus and the venous flow out of the villus are in directions opposite to each other, and that the vessels lie in close opposition to each other. Because of this vascular arrangement, much of the blood oxygen diffuses out of the arterioles directly into the adjacent venules without ever being carried in the blood to the tips of the villi. As much as 80% of the oxygen may take this short circuit route and therefore not be available for local metabolic functions of the villi. This type of countercurrent mechanism in the villi is analog to the countercurrent mechanism in the vasa recta of the kidney medulla. Under normal conditions, this shunting of oxygen from the arterioles to the venules is not harmful to the villi, but in disease conditions in which blood flow to the gut becomes greatly curtailed, such as in the circulatory shock, the oxygen deficit in the tips of the villi can become so great that villus tip or even the whole villus undergoes ischemic death and disintegrates. For this reason and other reasons, in many gastrointestinal diseases, the villi become seriously blunted, leading to greatly diminished intestinal absorptive capacity. Nervous control of gastrointestinal blood flow. Stimulation of the parasympathetic nerves going to the stomach and lower colon increases local blood flow at the same time that it increases glandular secretion. This increased flow probably results secondarily from the increased glandular activity and not as a direct effect of the nervous stimulation. Sympathetic stimulation, by contrast, has a direct effect on essentially all the gastrointestinal tract to cause intense vasoconstriction of the arterioles with greatly decreased blood flow. After a few minutes of this vasoconstriction, the flow often returns to near normal by means of a mechanism called autoregulatory escape. That is, the local metabolic vasodilator mechanisms that are elicited by ischemia override the sympathetic vasoconstriction, returning toward normal the necessary nutrient blood flow to the gastrointestinal glands and muscle. Importance of nervous depression of gastrointestinal blood flow when other parts of the body need extra blood flow. A major value of sympathetic vasoconstriction in the gut is that it allows shut-off of gastrointestinal and other splanchnic blood flow for short periods of time during heavy exercise, when the skeletal muscle and heart need increased blood flow. Also, in circulatory shock, when all the body's vital tissues are in danger of cellular death for lack of blood flow, especially the brain and the heart, sympathetic stimulation can decrease splanchnic blood flow to very little for many hours. Sympathetic stimulation also causes strong vasoconstriction of the large volume intestinal and mesenteric veins. This vasoconstriction decreases the volume of these veins, thereby displacing large amounts of blood into parts of the circulation. In persons experiencing hemorrhagic shock or other states of low blood volume, this mechanism can provide as much as 200 to 400 milliliter of extra blood to sustain the general circulation. So we have come to the end of the subject, general principles of the gastrointestinal function. Thank you very much for listening.